Hello, good morning to everyone. It's Paolo here, and we are going to start a Data Science webinar 101 in a few minutes. I want to thank you all for joining, and it's a pleasure to have this event. It has been quite a difficult way to arrive here, because we were planning to have the event not in this kind of situation, so not as a webinar, but as a live event. But then, because of the coronavirus pandemics, we decided to create this event in order to be able to have something all together concerning data science and bioinformatics. So I will now introduce briefly our webinar and our BioDataScience101 workshop. So, first of all, we want to thank DTU, SDC, the Science Danish Center, and Danish LTech for their support. And a little bit of presentations about who we are. This is me, Paolo, talking, and I'm associate professor at DTU LTech, Technical University of Denmark. And my expertise is everything that concerns the immune system, antibodies, T-cell receptors, vaccine development, and machine learning. Then today we have online Magnus. He is a new PhD student for working at Copenhagen University on the effect of mutations on protein stability in disease, and he's also an expert of machine learning. Then we have Andreas. He's also starting his PhD at the SDU, South Denmark University, and his expertise it's on single-cell sequencing. Finally, we will have later on Anders Pedersen. He's a professor at DTU LTech, and he's an expert of Bayesian statistics and evolution. Great. So let's start on the big questions of why we decided to have this this workshop. Last year, the Novo Nordisk Foundation published a research that it's concerning the state of bioinformatics in Denmark. It's called the Danish Bioinformatics Landscape. Now, these results are valid for all Europe and probably all the world, not only for Denmark. What we can see here is that if we focus on core bioinformaticians, so the people that develop methods, but also people that can use bioinformatics methods like applied bioinformatics and data science, we can see that in hospitals, in academia, and in the industry, there has been a big rise in the number of the people in the workforce that are required to have this kind of expertise. So we see, for example, for academia, there was a growth between 5 years ago and now of 62% in the requirement of this kind of expertise. And it has been estimated that in 5 years, we will have a even sharper rise of 87% for academia and 160% in the industry. So we can easily understand two things. First is that this is something that is extremely important in science today. Being able to work on big data, to develop methods that can be used to analyze this kind of data, machine learning, deep learning, and so on and so forth. But it's also clear that the universities will not be able to educate. So even if we have a sharp rise in the number of students that we can educate, and we already educate quite a lot, it's not going to be enough to cover the needs of the market. So our main idea was that we can also offer this kind of training and expertise to people that are already working, that are already employed. Really easy to approach any training in any kind of new expertise. So how are we going to do this? As I was telling you, it is clear that there is a barrier, some hurdles between what you want to learn and you. And this barrier can be, the fact that you work long hours, the fact that before learning data science, you need to learn how to program, or you need to install a lot of software that sometimes are not easy to install. You need to find the time to do that. You need, to deal with your family, for example, now with coronavirus pandemics, things are never easy. But we try to do our best in order to simplify the process. And our idea is that we want to ease your way up to the tasks and the things that we want to teach by, first of all, having a task-based approach. So we'll not teach you things starting from the theory, but we'll start with common tasks in the biotechnological industry. This makes things much easier because you will be able to remember things much better by doing rather than by reading or listening to lectures, right? This is something that we are very experienced at the TU. We do this quite a lot with our students, and it has demonstrated to be very effective. The second step is that we are going to use an interface that makes things much easier. That is the Jupyter Lab or Jupyter Hub interface. This means that you don't need to install anything on your computer. So basically, you just open your browser and you start programming. Of course, that this doesn't mean that things will be straight out of the box. You still need to learn the basics in programming in Python and statistics and so on and so forth. But we have removed the problem of installing software to learn how to do scripting in Python and all of these. And finally, we'll have an approach today teaching that I call back to base. This means that we start doing things, doing a specific task whenever we feel that there is a need to introduce some piece of theory or some technical skill. We'll deviate from our task and learn that. And this deviation can be as deep as you want. So you can, for example, today you will start using the notebooks, and you will understand that you want to learn to program better in Python. Then you will spend some time trying to learn the basics of Python. Then you will understand that you want to for example, study more machine learning. But at some point, you go back to the basics. That is the task, the initial task that you want to do. That is learn data science. That is learn how to do principal component analysis. So all these deviations, at some point, you have to ask yourself, okay, is this really meaningful for the original task that I was doing? If not, you go back to the original task. And this is a very effective way to learn things. Of course, as I told you, that's not, it's going to be easier, but it's not going to be simple, let's say. You still need to struggle a little bit in order to learn these kind of things. But we hope that this will be easy to manage for you. And the idea for today's webinar is to introduce kind of our approach. And of course, we want to introduce it by having you run through some notebooks so that you can actually feel if this is the right thing for you. In order to manage the expectations, the notebooks that we'll cover today are pretty long, and in some parts, they might be quite complicated. So I would say that if you have some expertise in Python and some understanding of basic biology, you will be able to complete 75% of the notebooks. So no worries if you are not able to finish the notebooks. If you have a kind of not such a great skill in programming, I would envision that you might be able to finish today 50% of the notebook. So no worries. Take things with a slow pace or with your own pace, and that would be the best way to approach this. There is also to notice one thing. The notebooks will not be offline after today, so they will stay online for 1 more week. So even though you might not finish things today, most of our people are in quarantine, so you still have 1 week to go through everything. Great. So that being said, I want to give you a short overview of the program today. We are now having this short introduction to the workshop. For the next 20 minutes or so, I will show you how to log into the notebooks on our servers and how to use Jupyter Notebooks. So some very basic introduction to Jupyter Notebooks. After that, I will give you a short presentation on the data set that we will use for the first module for 15 minutes, so 10 to 10:15. Sorry, there is a typo in the program. After these 15 minutes, you will be able to work by yourself on the notebook. We will be available for questions and for any kind of problems you might have. And at the end, at 11:15, all of us that have organized the workshop, so Magnus, Andreas, Hannes, and me, will go through the notebooks and give you some feedback or some additional idea on how it looks. And then we'll have a little bit of time before lunch for question and answer and feedbacks. And in the afternoon, it will be the same. So you will have some time between 1 and 2:15 to go through the second module. And after that, we'll have a wrap up and we'll get your feedback. The first module is focused on data exploration and modeling and how to do data I/O. That means reading data, but also filtering data and displaying data. The second module is more focused on data visualization, and we'll also do a little bit of statistical analysis and identifying sequence features that correlate with some kind of phenotypic effect. That's great. So what time is it now? It's 9:45. So let's move to the introduction to the Jupyter Notebooks. I will now share my browser. Now, all of you should have received an email that is containing a username and a password for connecting to the notebooks. Great. Just an internal feedback. Can you see the right tab in my browser? Okay, sorry for that. I'll have to just 1 second. Yeah, let's try this again. Super. Is it okay now? Can you see the BioDataScience101 webpage? Great, super. So you all should have received an email containing your username and password. Please be aware that usernames and password are specific for a given server. So you should use the server that you've been assigned. If you click on server, you see that you have two servers here. So just click on the one that you have been assigned. And in this case, I logged in automatically because the server was remembering my username and password. In the other cases, you have to put it in. I'll just log in from scratch. Just a second. Now I have to log out completely. Here. Yep. Here. Sorry, I couldn't find that. So here we are. I have my own username and password. You all should have yours. I can see that some people in the Q&A are having some trouble, and Magnus and Andreas will help you with that. And you can sign in. So if you have trouble logging in, just write this on the Q&A or raise your hand. If it's a technical problem, we will also see that and help you with that. And this is the main interface. So I'll give people a couple of minutes or 1 minute to log in if they have trouble. Great. So this is the interface that you should be able to see. You have on the left hand here a number of folders and scripts, and the ones that I have named module 0, 1, and 2 are the modules that we are going to go through today. And in the basic view, you should have something like this. So in order to start, you just need to double click on a module, or if double clicking doesn't work, you can also right click and open a module. This is as simple as that. And for people that are asking if some people are saying that there's no module 1 and module 2, no problem there. We will copy those. And in case, I will also show you how to retrieve them yourself. So here we have a gentle introduction to Jupyter Notebooks. The main idea of Jupyter Notebooks is that it's somewhere in between a Python script, and if people are experienced with R or with other software, it's kind of a command line, R command line interface. What does it mean? It means that, first of all, in a Jupyter Notebook, you have both commands, pictures, images, and code. And the code will be able to run in the Jupyter Notebook itself. Let's quickly go through the beginning. So you all should be able to click on a cell and that will become blue. And if you want to move, you can also press Shift-Enter and to go to the next cell. So let's try that and go through the very first few cells and get here to the double click me. You can also modify cells, of course. You just double click a cell And in particular, this is a markup cell, markdown cell, sorry. It means that it's a type of cell that you can use for commenting. You can write things with the markdown specific formatting style. You can also use HTML-based syntax, include pictures and all of these. Once you have finished working on it, you can either Shift-Enter or Control-Enter to see the result of your editing. For example, here I'm writing a second line that it's hello everyone. And you see you have modified the code. Now, this is very basic way to create comments. Let's move forward to the most important part. You have also cells that contain code. For example, here, 10 divided by 3. If now I execute this Shift-Enter, you will get the result for the cell. So the code in the cell is actually being executed. And you also have other types of cells, like the row cells, that are useful for notes or to write down commands in a quick and dirty way. If you now start editing a cell, you can change the type of cell from here. It is either code, markdown, or row. Now let's quickly go through the interesting part. We have here, for example, this cell. In this cell, we can see that there are some Python commands, but it will not be able to run because it's signed as a markdown cell. If now we switch and we make it a code cell and we again press Shift-Enter, it will actually execute the cell. So always remember this. I also would like you to notice this, that when a cell is executed, you see a small number next to it. This means that the cell is actually been executed and that the number corresponds to the order in which you have executed the cell. So for example, if now I print some other thing over here, you see that it has the number 3 because it has been executed after the number 2. Be very careful because this is a big source of mistake in Jupyter notebooks. They are great, especially if you want to explore data and do data science, but they can also be a bit misleading in some cases, simply because of the fact that you forget to run some cells and or that, for example, you don't run the cells in the way that you are supposed to. I will quickly go through the final part. The toolbar, you can do a lot of different things through the toolbar. You can save things, you can cut and copy cells, you can create new cells with the plus sign, you can execute cells. If you do something wrong and the Python gets stuck, you can also interrupt the kernel. No worries, because that will interact only your kernel, not all the other people kernel. So the other people will still be able to continue. And basically, you can almost do everything from the toolbar. Just spend some time, if you didn't do so before, just spend some time going through everything. Something that you should also be aware is that there is a lot of ways to interact with the notebook without using the toolbar. For example, you can delete cells, undo and redo, move cells, and select cells just by using your keyboard. For example, I mean, if you want to create a new cell above your one, you just select the cell, so you don't edit, but select so that it's blue, and then you press the letter A. You see you have created a new cell that you can edit. If you want to delete a cell, you make it blue, and then you press DT. Here it is. This is very useful, especially when you get a little bit more experience. Now, this is very important. There is a cell here. that is used, I mean, you don't have to understand what it does, but it's used to create some kind of helper tools through the notebooks. So let's remember to execute all cells. Now, final thing, Python. I understand that some of you might not be experienced in Python or have little experience. We will not go through the basics of Python because we believe that, I mean, this is completely another task. So if you want to become experienced in Python, that's not what we will provide you, the kind of expertise that we'll provide you. But also, you don't need to be very expert in Python in order to use the notebooks or to understand the concept behind the notebooks. What you need to know is that in Python, you might have variable. Variable are boxes in which you put values, and you do so by using this approach. For example, here I create a variable that it's number1 equal 10, number 2 equal 2, and then you can create a new variable that it's output, that it's the product of these two variables. You can execute it, and you see that it works. In general, every time that you execute a cell, the content of the last variable in the cell is displayed. You can also explicitly ask to display the content of a variable by using the function, the command print. Now, you can try and do some arithmetic calculations here. For example, I don't know, 3 minus 2.5 divided by 1.2, whatever. Another thing that it's important is that in Python, variables come in many different flavors. It means that there are variables that contain numbers, there are variables that contain integer numbers, there are variables that contain strings, and one very important type of variable is are lists. So, as the name suggests, lists are collections or, lists of values. For example, here we create a list that contains the numbers between 1 and 9. And you can also do operational list, for example, by doing the sum of all the numbers between 1 and 9. As you see, it's pretty straightforward. We will not spend time explaining all the type of variables that you have in Python for two reasons. One, it's because it's a very long discussion. Second is that one of the main things in Python is that the most powerful, probably the most powerful thing is that you will ending up using a lot of libraries, and each library has its own type of variables, type of objects, and it doesn't make sense to explain all of those. You will learn things on the way. Also, in some places, we ask questions. This is for you to check that you are on the right track. For example, you have a question that it's, what is the result of 123 by 234? You will have to write your own code, get the answer, and then you will have to write the answer here in this check answer module 0, module 1, and module 2. 28782, you execute, and you see whether you got the right answer or the wrong answer. So you get a feedback immediately if you're doing things in a correct way. Last piece of information, and this is usually something that it's a little bit hard for people that are not experienced in programming and in Python, is loops. We'll not use, in general, many loops throughout the notebooks, but there will be some. To give you a very quick explanation, a for loop, it's a way to repeat a given set of instructions more than one time. And it has this kind of syntax. For item in, for example, in this case, a list that contains the numbers 1, 2, 3, 4, 5, print item. Item will take every time one after the other the values in the list. So if we print, it will print one after the other, all the variable values in the list. Now, the list doesn't have to contain numbers. It can also contain strings, and it works all the same. Or it can even be a single string, and in this case, if we print the characters one after the other. And of course, the operations that you do are can be very diverse. You can, for example, calculate the square of each number in the list, and that's what you would get. Now, in the next few days, and if you will have time, I suggest that you go through the final part of Module 0 and trying to solve these small exercises, especially if you're not experienced with programming in Python. Also, we have included some information about two very important libraries that are used in Python. These two modules are NumPy and Pandas. And NumPy is a module that is used for numeric analysis, so to store and do operations on large numeric data sets, such as matrices or vectors. And Pandas is a module that is used to manipulate data. So importing data, filtering data, and exporting data, and all of these. Take some time, either now if you have time or in the next few days, to go through the materials that we have included. That being said, I will now go to the first module. I will quickly explain the type of data that we will use for the module, and then I will leave it up to you to go through the notebooks. So I'm opening Module 1 right now. And we can see that the learning objectives for this module are to use Pandas to import files and to filter the data, and to inspect and filter the data, to perform some analysis that are quite fundamental in data science, meaning normalization and missing value imputation, to transforming the data in general, to perform some descriptive statistics and some kind of data science trick in order to visualize complex data. Which kind of data we'll be using? It's data that is coming from this Nature Scientific Reports paper that is titled Exploring Experimental Cerebral Malaria Pathogenesis Through the Characterization of Host-Derived Plasma Microparticle Protein Content. Now, I don't want to focus too much on the biological content of the paper, but the main idea is that the authors tested the protein level in mice that are either healthy or after 3 days of infection with malaria, or after 8 days of infection. And what they want to see is that they want to answer to this question: are there markers of infection at day 8, or even better at day 3, so that we can detect the fact that a given individual has been infected early on, at a very early stage of infection. And the data that they analyze is obtained through tandem mass tagging mass spectrometry, TMTMS. This is a type of mass spectrometry in which proteins are chopped up in small fragments by using cryptic digestions. Then they are tagged to retrieve from which type of a sample they are coming from, and they are measured at the same time. It is important to know a little bit about mass spectrometry. One thing in mass spectrometry is that what you measure is the presence of specific peptides, but it's quite common that you don't see all the peptides present in your proteins. Only a few peptides, very specific peptides, will be identified by this method. For example, peptides that are not being able to be digested, so fragmented in a nice way, or peptides that are too much hydrophobic, too much hydrophilic, meaning that they have specific physicochemical features, will not be able to be identified. So it is expected that in this kind of data, we have a lot of missing values, and we have a lot of variability between samples just because of the experimental setup. Yes, this is a basic setup of the experiment. They had two technical replicates in which, so actually two biological replicates in which they had two mice that are not infected, two mice after 3 days of infection, and two mice after 8 days of infection. And these mice are, the proteins from these mice are then collected and pooled together, meaning that it's a single data set of proteins that is then being analyzed by mass spectrometry. And this is repeated 2 times. So you have 2 parallel experiments with different mice. So this is not a technical replicate, it's a biological replicate. It is important to understand that when we will analyze the data, we'll see both biological variability and inter-individual variability in our data. So the data set, the setup that was used in the experiment is kind of used to simplify and to diminish the variability that we observe, that it's not connected to the task that we want to solve. That means that we want to minimize the variability connected to technical or biological replicates or individual diversity. But this type of variability will always be present in the data. So we have one of the main tasks of data scientists is to be able to disentangle the variability that is connected to the task or the problem that we want to solve with the variability that is connected to the experiments or to the variability connected to inter-individual differences. Now, I will not spend more time on this. One thing that is very important for you to remember is that you need to run all the cells. So sometimes you might see errors in running the notebook. This might be connected to the fact that you might have not run some of the cells. So what you have to do is just Shift-Enter here, one after the other, all the cells, so that you are sure that you are running all the code that you need. Most importantly, run this first part in which we import all the libraries that we will use for plotting things. And this part that is used to get a better visualization of the results so that you can actually see all the plots and the graphs that you do directly in the notebook. And you will see that in some cases we will use color codes. I will quickly explain these. Green is for sections that are easier, so that we believe that people with not such a big expertise in programming and in the statistics, for example, should be able to perform the task that is described. Red cells are used for more complicated analysis. So try to do or to read those cells, have a feeling if it's okay for you to go through those, but also consider that it's nice for you to get a kind of a feeling of what you can do, even though you might not be able to understand 100% the content of that cell. It's kind of an interesting feedback for you to for example, to study that later, or to know that these kind of things exist. And then there are yellow cells that are used for notes, hints, questions. So also keep track of the cell colors. That being said, I will close it here and leave it up to you to go through the notebooks. If you have problems or questions, just use the Q&A, and Andreas, Magnus, and me will help you to solve your problems. Thank you, and we'll be back at 11:15 for our wrap-up of the module. Bye-bye.